Why are we teaching living cells how to do math? Seriously, why are we torturing these poor cells into doing something so unnatural? Silicon computers can already do this, so why reinvent the wheel? You could have used them to do very productive things such as making essential drugs, using them as test platforms to understand the deeper workings of biology, but math? Why? And another part of you might be wondering, how do we actually torture the cells into doing math? It doesn't even matter if there's no real use for this, there's just this itch you want to scratch. How is it that we humans need a brain to do math, but these cells just need a few of their buddies? There's just this part of you that wants to create man-made horrors beyond your comprehension, isn't there? Be honest with me. And well, today, I'm going to tell you that these viewpoints aren't in contrast at all. In fact, you'll see soon in this video, by playing with the building blocks of biology, we can use the insights gained from that to create some fascinating things that might even give computers a run for their money. So, I present to you how scientists managed to teach a glob of cells into doing the very basics of math, adding numbers, and why this man-made horror is actually the basis to the future of biology. This is what happens when you apply engineering to biology. Let's get started. We're teaching cells to add numbers, but how do we even add numbers using traditional logic gates? If you already know this, feel free to skip to the next part, otherwise this will be a pretty quick review. Computers work in base 2, 1 representing on and 0 representing off. In base 2, 1 represents 1 and 0 represents 0, duh. And just like in base 10, when you count beyond 9, you write 1, 0, or 10. But in base 2, when you count to 2, you write 1, 0. 3 will be 1, 1, and so on. There's nothing special about base 2 or base 10, they're just representations of the same thing. You can still add, subtract, multiply, divide, and so on. When we're adding numbers in base 2, we just take down the smaller digit as our sum and carry the higher digit to the next digit. And then we'll repeat this until we run out of digits, just like in base 10. To accomplish this, each digit requires 3 inputs. 1 from the first number, 1 from the second number, and a carry input from the previous digit. The output of this computation is the lower digit of the sum, known as the sum, and the higher digit called the carry, which the carry value can be plugged into the next digit, and we just compute until we run out of digits. This is in essence what a full adder does. It is achievable using this logic circuit over here. But how do we even teach a cell to perform this? It's not like it has logic gates built into it. And at this point, longtime viewers of my channel are probably smiling right now because cells are full of logic gates. If they weren't, then they won't be able to make decisions to save their lives. If there is okay food, then, well, digest the okay food, but when there's better food, then don't bother. In terms of logic, digest okay food when there is okay food, and not when good food is available. While logic can manifest in various ways in biology, in this video, we'll focus on the ones that can't be encoded in DNA. DNA holds the instructions that are transcribed into RNA, which in turn is translated into proteins. These proteins are like the workhorses of the cell. If the DNA is the code, then the proteins are the actual functions that get executed. Go watch this video for a deeper dive into this process. Of course, not all functions need to be executed simultaneously. The transcription stage can be controlled using something called transcription factors, which are often proteins themselves. And within this scheme, there is one feature that can lead to a domino effect that will cause the way we design logic circuits in biology to diverge heavily from their transistor cousins. See if you can spot it. Be sure to let me know in the comments. And now, at the transcription layer, we can start building the logic gates. First, let's create a sensor using a transcription factor that blocks transcription until a specific input chemical is detected. This serves as the basic buffer you've often seen in logic diagrams, allowing us to receive the input of the system. Next up, we can craft a NOT gate using a similar approach, but this time, the transcription factor enhances transcription instead of blocking it. To revert this, just add the specific chemical to that transcription factor, halting the transcription process. To build an AND gate, we require two transcription factors to join together to enable transcription. This way, the gate's output is activated only when both of the inputs are present. We don't have to build these ourselves. Most of the parts you see here are borrowed from bacteria, such as the buffer vanilla sensor as you can see here. So far, we've covered a sensor, a NOT gate, an AND gate. Now, let's move on to how we link these gates together. Remember, the transcription factors are also proteins, and we can use this to our advantage. Instead of having the output be any generic protein, we can make it another transcription factor, specifically the one that can activate the other gate down the line. And to illustrate that, here's a simple AND NOT gate. It only activates when there is the red chemical and not the blue chemical. Let's connect the light red sensor to the bluish pink transcription factor as the buffer, and let's connect the blue sensor to output the dark red transcription factor as a NOT gate. If the light red chemical is present, then the gate is lifted and we can make the bluish pink protein. But if you remember from earlier, that's not enough to activate our AND gate. Now, if the blue chemical isn't present, then we can make the dark red protein. And only now can we activate the AND gate. You can see that in this mechanism, we need both of these transcription factors to activate the gate. As for building an OR gate, we can connect the transcription factors in a clever way. By making the output of the two gates the same, when either of them are activated, the subsequent gate is triggered, creating the OR logic we desire for just a few extra components. You can see, if either the buffer or the NOT gate are active, the whole gate is active since they output the same light gray transcription factor. We can take this trick to the next step and obtain an extra AND gate for free. Here's how. This circuit over here works exactly the same as our old AND gate. Two transcription factors are needed for it to activate, but only with one twist. We can engineer one of the proteins to be unable to bind when a certain chemical is present, effectively introducing the NOT aspect and a free AND gate. Feel free to go back and look at these mechanisms again. There are a lot of moving parts. But the upshot is, the wires between the gates are transcription factors, and the gates themselves are encoded in the DNA sequence. However, there's one gate that is elusive in biology. That's the XOR gate. Building this is quite nifty in biology, but don't worry, we can just do the same thing as an XOR gate using AND or a NOT. So there we have it, the sensor, the NOT gate, AND gate, the OR gate, and a workaround for XOR gates. Great, we can start building the full adder now. This isn't that hard. Or is it? Imagine constructing a circuit with wires, but each time you need connection, you have to use a different color of wire. To make matters worse, there are only limited colors available. Sounds absurd, right? Well, that's precisely the predicament faced by biological logic gates. In biological circuits, each wire represents a transcription factor. Using the same transcription factor more than once forces connections between the gates that may not align with our intentions. What we used to our advantage to build the OR gate now works against us. And it doesn't end there. If we keep using more and more types of transcription factors, we'll eventually run out of available types. This limitation further compounds the complexity of our tasks. But that's not all of our worries. Even when using different types of transcription factors, they can sometimes crosstalk with one another. As a chemical system, unforeseen reactions can occur, causing interference and disrupting the precise functioning of our logic gates. On top of everything, the sensors we borrowed from bacteria were designed for the bacteria's specific needs. They didn't evolve alongside the human cells we're now working with. This mismatch introduces additional challenges in this already delicate process. All of these factors combined make biological computation seem almost impossible, and it feels like our hopes and dreams are about to be crushed to smithereens. How do we recover from this seemingly insurmountable blow? Is there a way out of this maze of limitations and challenges? In a remarkable twist, the answer lies within us, within the realm of multicellular organisms. Instead of relying on a single cell to perform all the functions, we can harness the power of multiple cells with diverse capabilities working together. In our case, we have 9 distinct cells, each equipped with their own logic circuit, collaborating as a full adder. By embracing this ingenious principle, we can swiftly address one of the 3 problems. Since each cell acts as a closed container, cells using the same transcription factor won't interfere with one another. This allows us to reuse the same color of wire multiple times. Additionally, we can borrow from biology and employ hormones to facilitate communication between cells. This enables us to selectively share the wires between the cells while keeping others contained within each cell. Who knows, maybe this approach can amount to something even more than adding bits. However, our journey is not without challenges. Building and optimizing a complex network of 9 different cells poses difficulties in testing and troubleshooting. How can we identify the cause of failure if it occurs? Drawing inspiration from engineering, we adopt a step-by-step approach, developing and testing smaller components individually, refining them until they function effectively, and then putting it all back together. Let's tackle the next obstacle: incompatible parts, sourced from different organisms. Surprisingly, we can modify the transcription factor proteins to incorporate segments from other proteins. It's akin to integrating a piece of code that resolves compatibility issues between the functions of the libraries. We can just take a human part and fuse it with a bacterial part. As mentioned earlier, it's crucial to test if this actually works. In this case, we encounter a persistent signal where the gate should be off. If the signal propagates through our circuit, it can be amplified and lead to undesired outputs. The solution to this challenge is kind of captivating. We encode a probability of self-destruction in the RNA form of the code, using something called a hammerhead ribozyme, a small RNA loop that cleaves the RNA into fragments. And you can clearly see the results improve. In biology, the code not only governs protein function, but can also serve as the function itself as well. This field of using nucleic acids as computers holds great fascination, and I might just delve into it in a future video. It's worth noting that the concept forms from the foundation of the intriguing RNA world hypothesis. Now, let's address the final issue of crosstalk. We can employ a technique resembling a confusion matrix, testing every chemical against every transcription factor. Even with just a few components, crosstalk occurs, such as the blue signal interfering with the yellow one. However, instead of avoiding it like usual, we can leverage the gate's ability to amplify signals by feeding the weaker signal into another gate. We can amplify it. And here are some results to boot. And that's it. That's all 3 problems solved, stemming from the simple principle of employing multiple cell types. We've accomplished our goal. Though, there are more challenges to address and solve, but this will make the video convoluted and really long, so the link to the paper for this work is linked in the description. And there you have it. That's how you build an adder circuit using cells. But now the question arises, why the heck did we just do that? Is there anything beyond just adding numbers using cells? All this serves as a proof of concept for cell-based therapeutics. Imagine cells capable of logically computing the production of specific protein drugs upon detecting certain diseases or markers. By exploring and constructing a simple system like this, we laid the foundation for developing more complex systems in the future. Without the essential intuitions gained through this process, building such intricate systems would prove immensely challenging. Furthermore, what we've done here offers a way to create simplified models of complex biological processes. Adopting a mathematical and engineering mindset, we construct toy models to gain deeper insights into the workings of the real thing. By playing with these models, we enhance our understanding and we can apply our knowledge to create new advancements. These creations, in turn, become the toys for others to explore. This iterative loop propels us towards designing better and more sophisticated bioengineering projects. Thus, our story begins with the quest to build computers using life. In the next episode, we'll be taking the leap from arithmetic to something even more profound. But for now, thank you immensely for watching.